It's a pleasure to be here. I want to admit I almost wasn't here. I thought my talk was tomorrow. I arrived here from America about 3 hours ago and was sitting in my room having a nice lunch and thinking, I'll get my slides together for tomorrow. And I got a call from Judith Eberhardt saying, why don't we have your slides yet? So I want everyone to notice I'm well shaved and showered and have slides and everything. I'm pleased to be here. I'm also disturbed by having been placed behind Kent Osborne and Matt Ellis, who I believe are doing some of the best work in our field. My talk, in my opinion, is modestly mundane, although probably important to those of us who are clinicians and want to know how to do, what to do with all the slides we just saw. And Matthew's next to last slide with the circle, I was sitting next to Chuck Perot, and he said this is the kind of thing that patients are going to come in and show us as clinicians, and I went like this. What are we going to do with that? And that's kind of what this talk's all about. Matthew, I wish I had that slide to show before I got started here because I think it's relevant to my talk. I believe that my title was given to me based on this movie. Some of you may remember Bill Murray and Scarlett Johansson being in what I consider one of the great movies of the last decade. And of course, now it should be co-starring, not co-starring, Dan Hayes, Genome to Bedside, Lost in Translation. And I don't know how lost we are, but I think we have gotten lost in many ways because in many respects, and certainly not the last two talks, but in many respects, we've fundamentally forgotten the scientific method when we apply all this new information to clinical research and clinical studies, and I think that's where we need to keep going. This is the usual disclosure information. I sit on a couple of scientific advisory boards, have some stock options, but nothing really relevant to this talk. So let's start out. Those of us who go to other countries and don't know how to speak their language, and that's most Americans, frankly, take with us a French to English dictionary or a German to English dictionary. I'd like to bring along here a science to English dictionary and try to define some of these terms that we all throw about, but I think are used semantically very carelessly and have resulted in a lot of confusion. Let's start out with the concept of personalized medicine. This is obviously what we all wish to do. Rich Schilsky, when he was president of ASCO in 2009, defined it based on what the Institute of Medicine of the United States had defined personalized medicine as getting the right treatment to the right patient at the right time using the right dose and the right schedule. If we're treating hypertension with beta blockers, that's probably not quite as critical as when we're treating patients with drugs that kill eukaryotic cells. And everybody in this room who wears a white coat knows that chemotherapy has a lot more toxicity than what the average doctor is using. And yet we really haven't been very personalized, but a lot of this next 3 days are going to be related to how can we personalize chemotherapy or how can we personalize targeted therapy like Matthew's talk. And so the point here is if we're going to personalize medicine, then we have to be just as worried about the science behind the biomarkers as we are the science behind the drugs. It's been amazing to me over the last 25 years in my career how we really wring our hands over trying to have high levels of evidence before we use new drugs. And yet markers come along and we use them to treat patients, and we probably treat patients incorrectly because we haven't worried about the science behind the biomarkers, and that's what the rest of this talk is all about. Indeed, actually, maybe we should treat everybody with everything, and some of you have seen me give this slide, but if a patient's willing to accept any toxicity for any benefit, then we should treat everybody with everything to ensure that we make sure everybody gets benefit. But that's not why we went to medical school. We went to medical school to learn how to assess risk and benefits and weigh them carefully. And if a patient is willing to forego some benefit to avoid some toxicity, and most are, then we should select our therapies very carefully. And that selection depends on our knowledge of prognostic and predictive factors and how good they may or may not be. And it also depends on our perspective. So one patient may think that reducing their odds of dying by 2% are worth the toxicities and cost, and another patient may not think that's the case. The same thing is true for those of us who are counseling our patients, and importantly, the same thing is true for societies. We may not be able to afford all these therapies, and we may have to make very tough decisions, and some of you are in countries that are already making very tough decisions regarding who should get what treatment and based on what criteria. I put up Peter Ravden's Adjuvant Online here as one example of a way to assess this. I think it's probably the best. Peter's gotten a lot of credit and should continue to get credit for this. He's worked very hard. So let's go on to tumor biomarkers. And again, many of you may have heard me say this before, but I'll say it again. Every fellow who trains at the University of Michigan hears this from me on just about a weekly basis, and that is that a bad tumor biomarker is every bit as harmful as a bad drug. Would you use a drug if you didn't know how it was mixed up, if you weren't sure what the concentration was, if you didn't have clinical data about how the drug might be useful, and especially if you don't have very high levels of evidence, usually from prospective randomized clinical trials, to determine how much efficacy and toxicity it might have? And of course the answer is no, you would not. And yet for some reason we seem to be willing to accept biomarkers without that kind of evidence. And that's raised a number of people to publish papers suggesting that we really need to discipline ourselves and take some order out of the chaos of the biomarker field in the same way we have with the drug field. That led a group from the American Centers for Disease Control to meet, led by Steve Teutsch, published in 2009, to articulate three terms that many of us had been talking about, but it was sort of like, you know, when you go to SeaWorld and you hear porpoises talk to each other, and you know those porpoises are saying something important to each other, but you can't tell what it is. And that's the way I think we were. We were talking to each other, but not really articulating very well. And they came up with three terms. One was analytic validity. Does the assay accurately and reproducibly measure what you just said, what you think it does? Matthew just hit on this, for example. He was talking about Sanger sequencing not being as accurate or reliable as you'd like it to be, but going to next generation sequencing. Those both, in theory, give you the same answer, but they're very different assays for the same answer, and you need to be sure you have analytic validity, including does the assay work in the kind of tissue that you actually have available, the pre-analytical issues as well as the analytical issues. The second is a term called clinical or biological validity, and that's just if you have a good assay, does it actually take one population and split it into two, and do those two populations look different? Do they look different either biologically, or more importantly, do they look different in terms of clinical response to therapy or outcomes? That does not mean it has clinical utility. Clinical utility means that the results of the assay have been shown with high levels of evidence to lead to a clinical decision that improves outcomes. That's a really high bar. There are probably 1,000 markers that have been published in the peer-reviewed literature regarding breast cancer over the last 2 decades, yet only 4 or 5 actually have clinical utility, actually have the kind of accuracy behind them and high levels of evidence that we're willing to use them to take care of patients. In my opinion, that's a shame. Many of those markers are quite interesting, but the studies have been done so poorly we can't figure out if they're good or not. And so that leads us then to how do we generate these levels of evidence. And I'm going to walk you through the results of a committee convened by the Institute of Medicine in the United States, in which a roadmap was developed for the determination of clinical utility of an omics-based tumor biomarker, which is mostly what we're talking about, although frankly these concepts apply to almost any biomarker. The origin of the IOM committee came about because of a really tragic set of circumstances at Duke University in the United States in which gene expression array signatures were developed that allegedly would predict whether certain kinds of chemotherapy worked or not. I think most of you know this experience. There were several papers co-authored by Drs. Potty and Nevins to suggest this was the case in really major journals: Nature, Science, New England Journal of Medicine, Journal of Clinical Oncology. There were several $1 million worth of grants awarded to Duke University to support this work. And most importantly, there were 3 clinical trials in which omics-based assays were designed and used to direct therapy. So patients were actually being treated with these assays. And these are the 3 trials that were up and running when suddenly, in America we have a saying, the wheels began to fall off the car. Suddenly there was an inability to reproduce these kinds of data in other centers. And I'm not going to go through exactly what happened in great detail, but it turns out that these assays were highly unstable and highly irreproducible. And so the trials were stopped, and the committee was put together to try to figure out what happened. Ultimately, what we said is we believe there are 3 stages of omics test development: discovery, test validation, and evaluation for clinical utility and use. And if you don't remember anything else from this talk besides the fact that a bad tumor marker is as bad as a bad drug, I'd like you to remember this figure. And this figure, going from left to right, is a good idea, the discovery phase, generation of a good test, the test validation phase, and then crossing that bright line, that yellow bright line, to evaluate as to whether that test does or does not have clinical utility used in the way that Dr. Teutsch and the CDC and EGAP initiative meant it to be used. Let's talk about discovery a little bit. That's the far left. You have candidate tests developed on a training set. Omics is usually some kind of exploratory analysis. Chuck Peru would talk more about that. He's the king of the world in that as far as I'm concerned. Matthew's talked just before me also, in which you do really unsupervised and in many respects undisciplined exploratory analysis to try to find things that might be interesting. That's the basis of science, in my opinion, is hypothesis generation. But then very quickly you need to confirm that that really is a good idea. How are omics tests different than development of new drugs? You need expertise in multiple disciplines. And again, I hearken back to Dr. Ellis's talk just before me. In my opinion, he's made more progress than anybody else in the field because he hasn't done it by himself. If you look at the people he graciously showed pictures of over and over again, he's a clinical oncologist. He has biologists. He has geneticists. He has statisticians. He has bioinformaticians. It's hard to say. I want to point out that biostatisticians and bioinformaticians are not always, in fact, often are not the same thing, and yet frequently one or the other is left out of these studies. Clinical pathologists to worry about pre-analytical issues. This work is hard to do, and it should be hard to do. Science is difficult, and in my opinion, Matthew's doing it right. The second is test validation, and now you've moved into the second box where you start out with a rough idea of what the test would be, but you're leading to something you're actually going to use to take care of patients. This is like going from a compound in your laboratory to a pill you're actually going to use in the clinic. So as you go left to right, you start out with a good idea, but ultimately you want to have a test that is developed on the training set, but then locked down before you cross that yellow line. This candidate test is evaluated on an independent sample set to at least get clinical validity, and then you're going to cross the yellow line to get clinical utility, and in each case, of course, you need both statistics and bioinformatics to get you there. And then finally, evaluation for clinical utility and use, and that's the far right-hand side of the box. And this takes you through getting a good idea down to actually determine who has clinical utility. Again, how are these different from drugs? First of all, at least in the United States and frankly in Europe as well, the regulatory oversight is a complete mess. It turns out there are two paths for bringing a test to clinical use. There are not two paths for bringing a drug to clinical use. You can either do it by getting FDA review, or you can just make a test in your own lab, and as long as you have a CLIA-based laboratory, which means that someone's come in and made sure everything's clean and you keep good records, and as long as you don't take that test across state lines, you can also use it in your own hospital without any FDA review. So let's go back to clinical utility. As the EGAP group described it, it turns out that when the FDA reviews a new test, they don't necessarily determine if it does or does not have clinical utility. So most of the tumor biomarker tests in our country have not been assessed for clinical utility. They've been a test for analytical validity, they've been tests for clinical validity, but not clinical utility. And furthermore, you can sell and use a test in our country, and frankly, I think probably in Europe, without FDA review by developing in your own lab. I think that's wrong, and several of us are trying to get that changed. Now, there are several ways to test the test, either prospective, retrospective studies, if you happen to have archived specimens from previously conducted clinical trials. I had the opportunity to write a paper with Rich Simon and Soon Paik in which we proposed a hierarchy of level of evidence and said that it's okay to use prospective, retrospective studies, but those samples should come from prospective clinical trials and not be studies of convenience. So now you're on this far left-hand side, having crossed the bright line, and you can take that down and get clinical utility, but it takes 2 or 3 of those kinds of studies to have the kind of level of evidence you'd like to have. If you don't have those archive samples, then you have to go into prospective clinical trials where the marker is the primary objective, and I refer you to 2 really great papers, one by Dan Sargent and one by Bruce Friedland, that go through different kinds of trial designs that one might use to test the test. There are two of these. One is the test does not direct patient management. So, for example, if you think that you're developing a prognostic factor, you can treat everybody the same way and then see whether or not your test finds the group that shouldn't have been treated versus not, that doesn't need more treatment. Or, for example, you can randomize people, but just you have to over-accrue to a study rather than just testing the drug in order to have enough power to actually then look backwards at whether or not your test identifies the patients most likely to benefit versus not. Or the other is the test does direct patient management, and in this case, you're actually using the test to direct how patients are treated. Examples of that right now are the TAILOR-RX trial in the United States, the MIND-ACT trial here in Europe, and the RESPONDER trial that's ongoing back in the United States, all of which are designed to test omics-based tests, investigate omics-based tests to see whether or not directing therapy with these tests results in better outcomes. So in summary, I'm going to say it again. A bad tumor marker is as bad as a bad drug. Regardless of the strategy you choose, the scientific method is still critical. Matthew and I have talked a great deal. I don't exactly know how we're going to take next genomic sequencing and apply the scientific method because, as he pointed out, the mutations that he and his colleagues are finding are so rare. But I think he's got the right answer, which is that if we find 3% of all breast cancers have a HER2 mutation, that's 4,000 or 5,000 people a year, we should be able to put those patients into randomized clinical trials. Regardless, the study design is important, proper controls are important, rigorous conduct and analysis. I believe you should have a written protocol with a date stamped on it before you touch a sample, just like you would for a drug, so that when said and done, if you get results that you like, you can actually believe them. And again, I'll say it, I don't know how we're going to translate next-gen sequencing into translational medicine, but I'm pretty sure Matthew does, and I'm looking forward to his teaching me how to do that. Having said that, I want to thank the people who ran the IOM report, and especially my colleagues who were on it. They worked very hard to come up with these things. It was led by Gil Lohman of my own university. He did a great job keeping all of us in order. And if you'd like to read this, it's on the web. You can download it for free. And if you're going to get in the field, I would strongly urge you to do so. Thank you very much.